So, this is going to be another fairly large chunk of information because this week we're getting into the other big key that lets us talk about the interesting aspects of how it is that organisms come to look the way they look. So, to be able to really discuss that, we've got to talk about reproduction and heredity. So, by the end of this lesson, you're going to be able to explain how organisms reproduce and how traits are passed on from parents to offspring. So, let's focus on reproduction, by which we mean how do you go from one cell to many cells? And it turns out that cells spend most of their time in this cycle. Right when a cell is first created, it is in its G1 phase. So it spends some time just growing and getting bigger. And eventually, it gets big enough that some chemical signals are set off, and the cell moves into S phase, which is when the cell makes a complete copy of its DNA. So that's when the DNA replication that we talked about last week happens. After that, the cell does a little more growing in its G2 phase until it reaches the point where it is ready to divide. And by division, we mean mitosis. So, mitosis is the most critical part of division. It is division of the nucleus of the cell from one nucleus to two nuclei so that the cell can then split apart. So, those copies of the DNA that were made during S phase are divided up. One set goes to one side of the cell, one set goes to the other side of the cell, so that each new cell ends up with one full copy of the DNA. And at the end of this process, the cell undergoes what we call cytokinesis, meaning it splits in half. So you started with one parent cell and end up with two identical daughter cells. Mitosis happens in 4 steps. So I'm going to take you through what happens in each of those steps. So the first step of mitosis is called prophase. And in this step, a couple of important things happen within the cell. First, normally in the nucleus, your DNA is just kind of loosey-goosey. There is a certain amount of structure to how it is organized within the nucleus, but it gets very highly organized during prophase. When you see those commercials on TV for various genetic testing services, you see shapes like the ones that are appearing on your screen. These are chromosomes. So that X-shaped structure is one chromosome. Each side of that structure contains the same genes in the same order, and each half of that X is a single chromatid. So 2 chromatids join together to make one chromosome. That's happening during prophase. Also during prophase, the nucleus starts to break down. So the DNA is going to be free to move out into the cell in the form of these chromosomes. And lastly, a structure begins to come together in the cell, the mitotic spindle, which is that red structure. It kind of looks like two pieces of macaroni stuck together. And it starts to send out microtubules, little protein fibers that are going to be very important as mitosis goes on. So there's this additional phase that you will see referred to in some biology textbooks and not others. So I wanted to address it because depending on where you go from here, you might end up someday in a science class with a teacher that tells you that there are 5 stages of mitosis. And the 5th stage that they're talking about is called prometaphase, which kind of takes some of bits of prophase and some bits of the next step, which is going to be metaphase, and squashes them together to make a 5th step for some reason. Personally, I think that the 4 stages make much more sense. So that's the way we're going to operate going forward. But I wanted you to hear this one so that if you hear it in the future, you know what it means. So let's progress right on to metaphase. And in metaphase, some major changes that happen are that the chromosomes line up. And they line up down this line that we call the metaphase plate. Just, it's enough to know that they form a straight line. And the centrosomes, those mitotic spindles that we're forming on either end of the cell, reach out those fibers to each of the chromosomes. And one fiber is going to attach at each side to the spot where the 2 chromatids meet, called the kinetochore. If it sounds like there's a lot of vocabulary in this, it's because it is. If you see it on screen, it's something important that I want you to know. If I'm not showing it on screen, you're probably okay with not remembering the exact vocab word. Just know that each of those sister chromatids gets a lovely protein fiber attached to it, and they line up down the middle of the cell. And in anaphase, we start to see that those 2 sister chromatids separate. Remember that each side of that chromosome, each one of those chromatids, has the same genetic information on it. So one copy gets pulled towards one end of the cell, one copy gets pulled towards the other end of the cell, dividing them up so that what you're going to end up with is one full set of DNA on each side of the cell. This is telophase. And this is where that entire process wraps up. So the critical things that happen in telophase are basically the opposite of what happened in prophase. So the chromatids that were pulled to each end start to unravel, and a new nucleus starts to form at each end of the cell, and that mitotic spindle structure starts to break down. So telophase is just undoing everything that happened in prophase, one set at each end of the cell. So by the end of telophase, what you've got is the formation of 2 new nuclei. After this, remember, the process of cytokinesis happens. So cytokinesis just means you started with one cell, it's going to pinch itself off so that now you have two separate cells, each with its own nucleus, with a full copy of the DNA. This is one way that cells can make more cells, which means that it's a form of reproduction. Specifically, we call mitosis asexual reproduction. Anything that means that you are starting with a single organism and that single organism is capable of making offspring is going to fit under the label asexual reproduction. This includes mitosis, or when we're talking about reproduction, we call it binary fission, meaning a single-celled organism splits into two similarly sized single-celled organisms that are identical to their parent. But that's not the only option. You can have fragmentation. Think starfish. The arm of a starfish falls off and is capable of regrowing another full starfish. And that starfish would be identical to the original that lost its arm. You can have budding, and for budding, our classic example is yeast. If you look at live yeast under the microscope, and we would have actually observed this if you had been in the school building with me at this point, but I'll put a video in so that you can see this happening virtually. A parent organism pinches off a smaller organism that it grows for a while as part of its body. So yeast do this all the time. We can have vegetative propagation, which is similar to fragmentation, but specifically happens in plants. So a new organism can fall off of the parent organism. We can also do this as humans. You can take a clipping from a plant and grow a whole new plant from it. And finally, the weirdest one on the list for sure, parthenogenesis. There are some species that are capable of doing something that looks like the other kind of reproduction, sexual reproduction, which we're going to talk about in a second, but with only one genetic contribution. So some species of lizard, for example, can do this thing where they create an egg and it looks like the reproduction of any other lizard, except that there are no male lizards. It's one sex of lizard that is capable of reproducing completely by itself. But not every organism is capable of or really wants to reproduce its species in this way. And we'll talk in a second about the advantages and disadvantages. But there's another way. So the other option is sexual reproduction. And that one gets a little complicated. Think about it. The DNA is what's the problem. You've got to have exactly the right amount of that, or things start to go wonky. So when you have two parents and you're trying to incorporate a genetic contribution from each, you want an organism that still ends up with the normal amount of DNA, whatever that is for the organism. So you have to have some kind of reproductive mechanism that includes cutting the normal amount of DNA in half so that then you can combine half the DNA from one parent and half the DNA from the other parent to get one complete organism that has the correct amount of DNA. And that process is called meiosis. You're going to notice that as we go through the steps of it, it's like doing mitosis twice. So the end result, you start with one parent cell and end up with 4 daughter cells, twice as many as mitosis. And those 4 daughter cells are different from the parents. Normal cells, when they go through mitosis, end up with 2 copies of each chromosome in a cell. So we call them diploid because they have 2, di, the prefix 2. So 1 copy of each chromosome in each cell is the end result of meiosis. We call these cells haploid because they have half of the normal amount of DNA that a body cell should have. So let's pass through the steps. And a lot of them are going to look very similar to mitosis. They even have the same names. But because we do 2 rounds, you have a prophase 1, and later on you'll have a prophase 2. So I'm going to assume that you were paying attention a second ago when I talked about mitosis, and just take you through what's different in meiosis. So in meiosis, the first time we enter into prophase, we add an additional step. After those chromosomes condense, they line up in pairs. The one you got from one parent and the one you got from the other parent. And something unique happens. See, chromosomes always work out so that, say, the gene that codes for the lactase protein is always going to be in the same spot on the chromosome. So these 2 chromosomes that have the same genes on them, just from different parents, do a process called crossing over, where they swap genetic information from one chromosome to the other. So the lactase protein that I got from my mother could swap places with the lactase protein gene that I got from my father. So that now this chromosome over here is mostly the DNA that I got from mom with a little chunk of DNA that I got from dad. And this one over here is mostly DNA from dad with a little chunk that I got from mom. This process is totally random, and it's incredibly important because of reasons that you will see soon. So next we move into metaphase. In normal metaphase, in mitosis, the chromosomes all line up one on top of the other. In meiosis, in metaphase 1, those 2 identical chromosomes line up side by side. And those 2 identical chromosomes are what's going to be separated in anaphase 1. So those 2 identical chromosomes separate, new nuclei form around them. This bit looks exactly like mitosis. And just like in mitosis, the cells are now going to split apart in cytokinesis. So far, we've done half of meiosis. We've made 2 daughter cells, and each cell has 1 chromosome from the original pair of identical chromosomes. Now we run the same thing over again. And part 2 of meiosis looks identical to mitosis. So we do weird mitosis for 1 round, and then normal mitosis for 1 round. Put them together, and you have meiosis. So once each of those 2 daughter cells runs meiosis part 2, you end up with a total of 4 new daughter cells. Each of those cells has 1 single chromatid out of the original pair of chromosomes, And we call these cells gametes. In humans, those gametes are either sperm or egg, depending on what kind of human you got sitting in front of you. But regardless, they have the same basic genetic makeup where they have half of the amount of DNA that a normal cell would have. So then, in sexual reproduction, one gamete from one parent and one gamete from the other parent combine to make a whole individual that has half of its genetic code from one parent and half from the other parent, which is a mixture of what those parents got from their parents, which is a mixture of what those parents got from their parents, and so on. How do you know whether a cell is going through mitosis or meiosis? If it happens in any old normal body cell, results in two offspring that are identical to the original cell that you started with, and makes a diploid cell, meaning there are two copies of each chromatid, it's mitosis. On the other hand, if it only happens in reproductive organs, results in four offspring that are not identical to the original cell, and makes a haploid cell, meaning one copy of each chromatid, it is meiosis. So, why bother having two kinds of reproduction? Well, because there are advantages and disadvantages to each. So, the kind that we just talked about, sexual reproduction, the advantage is variety, which is great if you want to be highly adaptable to your environment. And it's also great if you want to have better chance at surviving certain kinds of calamities like diseases. The disadvantages are, comparatively speaking, it takes a lot longer to grow a new organism via sexual reproduction than via asexual reproduction, which limits your population size. So, on the other hand, asexual reproduction doesn't require any kind of specialized cells or machinery. It's fast, and you can make a large population for a very limited cost of energy. But on the other hand, the trade off is adaptability is limited. You're pretty reliant on random mutations for hoping that you're going to produce something helpful that will let you survive in your environment better. And if a disease causing agent figures out how to exploit some genetic quirk, you can end up losing a lot of members of the species very quickly because they're very genetically identical.
All right, I've told you before that the people in my family come in exactly two patterns, right? We either look like this or we look like that. And yet somehow we are all related to each other, despite the fact that some of us look like this and some of us look like that. And the key to why it is that we can all be blood relatives and look as different as we do is all down to genetics. So I brought you into the Smith family photo corner to give you a little more context and to show you how it is that we can end up looking like there are exactly two patterns of us. So this is my lovely family, my mother and father, and my brother and sister and I. And you can see that Mom and my brother and Noah and I all look eerily similar. And Dad and my sister Rachel look eerily similar. And if we come over here, these are my grandparents, my mom's parents. Mom and I both look an awful lot like that guy. But you can start to see where my sister might have come up with some of the traits that make her look different from us. And on the other hand, it's pretty easy to see why my dad looks the way he does when you take a look at his parents. So the study of genetics is all about taking all of these people that are on the shelves in front of me and figuring out why it is that some of them look one way and some of them look another way.
So this cool looking dude is not related to me, but he is related to the topic at hand. This is Gregor Mendel, who is considered to be the father of modern genetics. Gregor was a monk, and as part of his duties, he was a gardener at the monastery where he lived, and he made some interesting observations about the plants that he grew in his garden, specifically pea plants. So, out of his observations, and I'm going to show you what he observed in a second, he came up with this idea that there are different versions of whatever codes for traits. At the time when he was doing his work, we didn't yet know what it was that passed on traits from parents to offspring. DNA wouldn't be discovered for another almost 100 years. But he understood that parents pass on traits to offspring and that there are different versions of those traits. And he discovered that some of those versions are more likely to show up in the offspring than others. So we can predict how these versions are inherited. This idea is called Mendelian genetics, and it works really well to describe what happens when you have a trait that is coded for by a single gene. For example, Mendel worked with pea plants. And if you've ever seen a pea plant before, you know that they have flowers, much like with other plants. From the flower, then comes the fruit. So it turns out that in Mendel's garden, there were two colors of flower on his pea plants. Some of them, same species, produced purple flowers. Others, same species, produced white flowers. So Mendel's experiments involved breeding pure strains of plants where if you got seed from the plant, those seeds would produce purple flowered plants every time. And plants where if you got seeds from a white flowered plant, they would produce white flowers every time. So that he knew that their traits were pure. That you had a purple colored plant that would only make purple offspring, a white plant that would only make white offspring. And then he would crossbreed those plants and see what he got. And then he would crossbreed the next generation and see what they got. And what he worked out was something like this. As long as the plant has at least one copy of the purple gene, it will have purple flowers. If the plant has two copies of the white gene, it will have white flowers. So he labeled these traits dominant and recessive. In pea plants, it turns out that the purple flower gene is dominant. The white flower gene is recessive, meaning it will not be expressed if there's a purple gene. So, if you breed two plants, each of which has one dominant and one recessive gene, we can predict that about 75% of their offspring will have purple flowers because they'll get at least one copy of the dominant gene from each parent. But about 25% will happen to get the recessive gene from each parent. So 25% of those plants will come out to be recessive and show the white flowers. Now, this classic Mendelian inheritance doesn't work that well for more complex traits. For example, let's talk about humans. Let's talk about height. Height is coded for by so many different genes that we don't really understand the mechanism for how height is inherited yet. And it turns out that there aren't many true Mendelian patterns in humans that we can study. As a student in high school biology, I learned that things like whether or not you have a widow's peak in your hair, like I do, or whether or not you can curl your tongue like this, are single trait Mendelian genes. And it turns out they're not. None of the things that I learned about in high school as in humans really turn out to act like what Mendel studied in his pea plants. But here's a human example that does: lactase persistence. So my mother can digest milk. My father can digest milk. My siblings can both digest milk. As we have discussed several times before in this class, I am lactose intolerant. We know that lactose is digested by the enzyme lactase. We know that an enzyme is a protein. We know that proteins are coded for by DNA. So somewhere there must be a gene whose job is to make lactase. And it turns out that there is a single gene that is responsible for lactase, and that there are two different versions of that gene. We call these different versions alleles. And the two alleles of the LCT gene that codes for lactase, the dominant gene, turns out that it maneuvers the DNA a little bit differently and makes it easier for the proteins necessary to transcribe that DNA to get in. So in the dominant version of the gene, transcription happens more easily, more mRNA is produced, and more lactase protein is produced. In the recessive version of the gene, the DNA is coiled slightly differently, makes it harder for the transcription proteins to get in there and transcribe that gene. So if you have the recessive copy of the gene, you get less mRNA being produced, which means you make a lot less lactase than normal. So if you have at least one copy of the dominant version of the allele, you make enough lactase that you're able to digest milk just fine. But if you have two copies of the recessive gene, you are lactose intolerant. So I can work out, based on that information, that my parents and my siblings all have at least one copy of the dominant allele. So my father inherited from his parents at least one dominant LCT gene. And my mother inherited from her parents at least one dominant copy of that gene, because they were able to pass on those genes, because they had them, to my siblings. So both of my siblings must have at least one dominant copy of the gene. But I can also work out that my father from his parents inherited at least one copy of the recessive version of this gene. And my mother inherited from her parents at least one copy of the recessive version of this gene because they were both able to pass on that recessive allele to me. So I know that my mother has one copy of the dominant gene, which, as a convention, we always represent dominant alleles with a single capital letter. In this case, I picked L for lactase. And my mother has one copy of the recessive gene, which I represented with a lowercase l, leading me to have a genetic profile that looks like lowercase l, lowercase l. I have two copies of the recessive allele. My siblings, on the other hand, could have two copies of the dominant allele, capital L, or they could be like my parents and have one copy of the dominant allele, capital L, and one copy of the recessive allele, lowercase l. Genetics is fun, right? So this week, we're going to really focus in on the reproduction aspect, but I wanted you to have that intro to heredity because next week, we're going to be all about it and all about how traits are passed on from parents to offspring. So next week, we'll dive into Mendel's research a little more. We'll talk about that whole capital letter, lowercase letter thing. We will make some of those Punnett squares that you saw with the pea plant and do some practice at tracking a gene through a family tree. For this week, focus on making sure that you really understand those two processes of mitosis and meiosis. And do good work. Be good, be safe, be kind. Don't do anything I wouldn't do. Wash your hands, and I'll see you next week, if not before.